the various type of glands, merocrine, apocrine and holocrine, because recently some questions are coming. We've got to discuss the three types in this diagram, and let us first focus upon salivary glands. The merocrine variety. Now if you say merocrine, how do you know salivary gland is merocrine? Because what I find is that secretions are coming, but there is no loss of cell membrane. So without loss of cell membrane, some secretions are coming, and that is called as merocrine type of secretion, like seen in salivary glands. And what about the other one, which is apocrine? Apocrine is a sweat gland. Actually, sweat gland is a modified, this mammary gland is a modified sweat gland. Now, if you say mammary gland is a modified sweat gland and it is apocrine variety, how will you know that it is apocrine type of secretion? Because here, apical cell membrane is lost and cover the secretions. And what about the third one, which is holocrine? Holocrine, like sebaceous gland in the skin. Holocrine, like sebaceous gland in the skin, whole of the cell membrane is lost and become the secretion, the sebum. So you're telling that it is called holocrine since whole of the cell membrane is lost and entire cell will become secretion. You mean to say nucleus will also break down? Yeah, nucleus will also break down, entire cell organelles will break down and become the secretion, like here it is sebum. Let us look at them histologically, because this is schematic diagram, histological diagrams. This is histological diagram. So this is merocrine, this is apocrine, and this is the holocrine. Yes. Let us focus upon merocrine. What did you say for merocrine? How do you identify merocrine? Merocrine is also called as acrine. So whether you say acrine or you say merocrine, the thing is, this is one gland here. I'll mark it. This is one of the gland. This is another gland. So what do you want to tell here? What I want to tell is, actually, there is no loss of cell membrane. There is no loss of cell membrane, and the secretions are coming up, but without any loss of cell membrane. But how do I know that there is loss of cell membrane? That you can see here, compare the two. And what will you find there? What we find there is, see, there is some loss of cell membrane, and you can see that the secretions are covered by the cell membrane itself. So you mean to say these are the secretions which are covered by some part of cell membrane and loss of some apical cell membrane. Apocrine, yes, there is formation of blebs. Blebs are formed. And you can draw a diagram yourself, but this is apocrine type of secretion. And what about the holocrine? In holocrine, we have discussed, like sebaceous gland, the peripheral cells are intact, but towards the duct, there will be loss of cell membrane, loss of nucleus, and become secretion. So you mean to say that periphery, the cell margins are intact? Yes. But towards the duct, the cell margins are being lost and the nuclei are also lost and everything becomes sebum now. Yeah, that is the point here. Now we are going to draw a diagram which is going to compare the three types, merocrine, apocrine and holocrine. Let's do that. If you are having a gland where the cells are giving the secretions, but there is no loss of cell membrane, then you will tell that it is acrine, which is also called merocrine. This is the point. So secretions are coming, but without loss of any cell membrane, that is merocrine or acrine. What about the other one? Other one we have discussed that this is a gland now, and the cells are having apical loss of cell membrane, and that secretion, whatever secretion was there, is being covered by the cell membrane itself. So this is which variety? Actually, this is what we are calling as apocrine variety. And how do you know it is apocrine? Because there is loss of apical cell membrane. So apical cell membrane is lost? Yes. And it is covering the secretion. Yes, that is the point. So apical cell membrane is lost and covering the secretion. That kind of appearance is apocrine. What about the holocrine? Holocrine, you can take example of a hair follicle. This is hair follicle. And if that is hair follicle, then which gland is associated with that? Say this is the sebaceous gland. This is the duct of sebaceous gland. If that is the duct of sebaceous gland, then near the duct you will see that there is loss of entire cell membrane. See, periphery, the cells are intact with membranes, but towards the duct, the nuclei are lost and the cell membrane is also lost. So that entire thing is becoming sebum. So periphery, the cells are intact, but towards the duct, sebum is formed, and there is a loss of cell membrane and nuclei. Everything is, becoming sebum. That is how you identify the holocrine gland. So that was the diagram for holocrine gland. Now we got a question. Which of the following is a holocrine gland? Immediately you will get your answer. There is no problem with that. But I have got more questions for you. Say you will tell my answer is sebaceous gland. Fine. Agreed upon. But what about the sweat gland? There are two types. And what are the two types? Could be merocrine, apocrine. Which is more common? Is merocrine. And which is less common? The apocrine. Now, if you say apocrine is less common, can you tell me where they are found? Yeah, they are found in the axilla region and perineum region. Now, if you say they have limited distribution and in the axilla region, perineum region, perigenital area, can you tell some other places where you have apocrine? Sweat gland. You already know two of them. You remember? Apocrine sweat gland, modified apocrine sweat gland. So one of them was ceruminous gland, wax gland, modified apocrine sweat gland. What was the other? Modified apocrine sweat gland. That was the mammary gland. So mammary gland is also a modified apocrine sweat gland. What about the pancreas? Pancreas is merocrine. Actually, pancreas has an exocrine component, and that exocrine component is merocrine variety. By the way, what is the answer to the question? Answer to the question remains the same. See, merocrine is pancreas, but the answer remains sebaceous gland because the question is on holocrine. So keep that answer. Now, when we talk about the glands, serous, mucinous and mixed also come into play. How do you differentiate between serous and mucinous? Serous is watery secretion, thin watery secretion. And what about mucinous? Thick, viscous secretion, thick and viscous. So where do you see the serous gland? Maybe parotid, salivary gland. And what about the thick viscous? Mucinous, maybe sublingual, salivary gland. And what about the mixed seromucus? If you say mixed, which is seromucus, give the example because that is a question. It is the submandibular salivary gland. Though you will find that this is not precisely correct because sublingual is also mixed type of gland, though majorly it is mucinous. So the point is that sublingual salivary gland is majorly mucinous, but it can be seromucus. But if somebody asks you mixed salivary gland, I'll prefer my answer as submandibular salivary gland. Let us look at the details now. See, this question is asking serous demi-lune. And where do you find them? Serous demi-lune are feature of seromucous gland. And where is seromucous gland? Can be sublingual, but which is the better answer? The better answer is submandibular salivary gland. Why did you take this answer? What is parotid? Parotid is serous. Then what is sublingual? Chiefly mucinous. We want to discuss these details, but keep your answer as choice number B and proceed further. As you're proceeding further, you'll find that if you are talking about parotid salivary gland, it will give thin watery secretion. We'll magnify the diagram and see that how do the serous gland look like histologically. If you're talking about the sublingual salivary gland, which is under the tongue, it is mostly mucinous type of cell. They have light stain and some foamy appearance due to mucinous secretions. Light stain, foamy appearance. And what about the submandibular? Submandibular is under the mandible and it is seromucinous. Some serous gland, some mucinous gland, and that is where you can have a serous demilume also. So let us talk about that as well. But at this moment, I am looking at submandibular salivary gland. So if this is a submandibular salivary gland, that means it is seromucinous? Yes, because I see this is a serous gland. This is another serous gland. This is another serous gland. But then this is a mucinous gland. This is a mucinous gland. This is a mucinous gland. Actually, mucinous glands are identified by the fact that they are lightly stained and they have foamy appearance due to mucinous inclusions, whereas this pink stain or darkly stained is serous gland. This is serous gland. This is serous gland. These are serous acini and these are mucinous acini. Can you see one more thing here? This is once mucinous acinus, and over that you have a cap of serous gland, a cap of serous gland. You see, this is a cap of serous gland. This is a cap of serous gland. So you are having a cap of serous gland on mucinous gland. Yes, this is called serous demilume. Serous demilume. Seen in mixed type of gland, seromucinous gland, like submandibular salivary gland. You see here, there is a serous cap on a mucinous gland. How do you know that it is a serous gland? I am telling you, this is the cap and this is the gland here. Which gland? The mucinous. And what is this? See, this is serous demilume. So this is a mucinous gland and that is serous demilume. Yes. You mean to say that this is a mucinous gland and that is a serous demilume? Yes, serous demilume. This is a serous demilume. How do you know that this serous demilume? Because these cells are cuboidal and they are dark stained, eosinophilic. So they are dark staining eosinophilic like this one. Yes. Serous glands are dark stained eosinophilic cells. Then what about the mucinous? Mucinous are light stained and they are columnar. How do you know it is columnar? Because oval basal nucleus. Oval basal nucleus, you mean to say columnar? Yes. Height is more than the breadth. So height is more than the breadth and then there's an oval basal nucleus, so it is a columnar cell, so it is some mucinous acinus. Yes, this is mucinous acinus, but that is central spherical nucleus, central spherical nucleus, central spherical nucleus. So this is cuboidal, this is serous acinus. So where do I see mucinous? Mucinous you already have seen. This is mucinous, this is mucinous. That is how you identify, histologically. These slides are given to you in the exam, in the computer. You have some 20 seconds. Identify the slide, 20 seconds. You have to train your eyes by practicing more and more of MCQs.